APS's Science Policy Committee decided to focus on the topic of reproducibility, honestly because we're scientists. We're curious people, we're always asking the next question. As physiologists, we want to understand how organisms work and help treat diseases and really continue to ask those questions. And those are building blocks. And each scientist builds on each other's building blocks. And the idea that there's cracks in those building blocks, such as challenging the idea of reproducibility, is something we need to address. There's a lot of different reasons that things can't be reproduced. So therefore, disclosing how experiments were done is really key. If scientific research can't be reliably reproduced, there's so many implications. The trust in the scientist is going to erode. NIH and funding agencies give billions of dollars to scientists to help better create cures, etc. And we want to make sure that money is well spent. Animals sometimes don't always reproduce into human studies. The first thing to remember is an animal's not a human. And a lot of times we develop animal models to look at very specific questions, not at the disease as a whole. So there's a lot of different ways that it can't reproduce. We're not identical. So how do we improve that bench to bedside relationship? And as I said earlier, it's all those building blocks. And if we can improve those building blocks and make sure they're valuable and that those lessons are learned, that improves that ability for the next scientist, which then in turn is kind of like a cascade effect and gets from research to bedside much quicker and better and helps provide those opportunities and those cures in a more efficient and better manner.